Our question today is, what really happens during cellular respiration? Like, we know it's important because we're spending 2 weeks on it, but why is it important and what's happening? The first thing that happens is glycolysis. Now, our learning goals are going to be the things that we want to know for exams and quizzes. And for glycolysis, we want to summarize what happens in words and list the inputs and the outputs. Glycolysis literally means breaking sugar. So that's what we're going to do in this first step. Now, this is the only step that occurs in the cytoplasm, and it's the only step that can occur if there's no oxygen present. Glycolysis starts with a glucose, which is a 6-carbon molecule. And we are going to add 2 phosphates to the end of this molecule. To do that, we have to use 2 ATP. We're going to rip that last phosphate off and stick it to our molecule, creating 2 ADP. This 6-carbon molecule is then split into 2 3-carbon molecules. Another phosphate's added to the ends to keep them stable. And then we remove both phosphates, and as we remove both phosphates, we can add them back to 2 ATP, creating 2 more ATP, and we also can harvest some electrons. NAD is like an empty taxi. It's ready to pick up electrons and take them somewhere else. Now, I know people don't really use taxis as much anymore, so maybe you can think about this as Uber or Lyft. So this is a person waiting for a ride. And then once you have an electron that you're carrying, the molecule becomes NADH, which is a full taxi. This is full of electrons, ready to take electrons to another step. Our end molecule is called pyruvate. Pyruvate is a 3-carbon molecule. And we end up with 2 of them. Summarizing words, what happens? Glucose is converted into pyruvate. In the process, we produce 2 full NADH, that's one of our outputs, and 4 ATP, another one of our outputs, except it costs 2 ATP to do this process, so we only make 2 ATP net. We make 4, but we use 2, so we only get 2 total out of it. Now let's take that pyruvate and go over to the mitochondria. So the mitochondria has its own learning goals. We want to identify the parts of the mitochondrion, which is singular, and then we want to explain how the structure of a mitochondrion, or multiple mitochondria, which is plural, relates to its function. Here we have a diagram of a cell. We've highlighted the mitochondria, then we're zooming in on this mitochondria in this diagram. This image on the right is an actual mitochondria, which is pretty cool. This was taken with a scanning electron microscope, and this is a mitochondria cut in cross-section. So on the outside of both these images, you have the outer membrane, that is C. Then you have the space between them, which is like this little white line between the 2 dark lines. That is the inner membrane space, that is B. Then you have the inner membrane, which is D. The inner membrane is folded inward, and when we draw it, we always show it kind of as like wiggles or waves. In reality, it goes all the way across almost, so really drastic infoldings. These infoldings are called cristae, which sounds like a first name to me. Hi, cristae. So cristae are the infoldings of your inner membrane. The space inside the inner membrane here, which is being shown by F, is the matrix of the mitochondria. Then G is showing you a little bit of DNA, because mitochondria have their own DNA separate from the DNA in your nucleus. And this one, whose label got cut off, this is pointing to some ribosomes, because your mitochondria also makes its own proteins. So mitochondrial DNA, mitochondrial proteins are separate from the rest of the cell. We labeled the parts. Now let's talk about why the structure is important to the function. This little mitochondria got its second membrane, its outer membrane, in the endosymbiotic event that you learned about before. And these infoldings, the cristae, are really important because they increase the surface area of the inner membrane. So the cristae, the structure, are related to the function of the mitochondria, which is to produce ATP. We want a lot of inner membrane because the inner membrane is where we're going to find the electron transport chain and the enzyme ATP synthase. And both of those components are important to making the bulk of our ATP. Here we have the citric acid cycle. You may have heard it been called Krebs cycle before. I'm going to use the term citric acid because we're kind of moving away from calling things after people's last names. So citric acid cycle. Our learning goals for this process are to summarize in words what happens, list the inputs and the outputs, and follow the loss of carbon atoms. We're going to start up here with pyruvate, which we made during glycolysis. And pyruvate is not the starting molecule. Pyruvate needs to be converted into acetyl-CoA. So this process to convert pyruvate into acetyl-CoA is called pyruvate oxidation, and some textbooks show it as its own step. Not always, sometimes it's included in the citric acid cycle, like what's being shown here. So we're going to take this 3-carbon pyruvate, we're going to crack one of these carbons off. See how acetyl-CoA only has 2? And we're going to release that carbon as carbon dioxide. We're also going to harvest electrons in NADH, our electron taxi. We're going to add an enzyme called CoA, leaving us with the molecule acetyl-CoA. Acetyl-CoA can then start the Krebs cycle. When you take acetyl-CoA and you bind it to this molecule called oxaloacetate, which is just such a rad word, go ahead and say it. Oxaloacetate, you create citrate, which is where the term citric acid cycle came from. Now, I don't think that you need to know the rest of these molecules' names. I'm not going to have this graph or this figure on the exam with this one missing and ask you what it is. But acetyl-CoA, oxaloacetate, and citrate are important. So acetyl-CoA has 2 carbon, which are left over from that glucose you ate. Oxaloacetate has 4, citrate then has 6. So you have this 6-carbon molecule here. This 6-carbon molecule is going to go through several different conversions. And in the process, you're going to pop off 2 more carbons, which you're going to release as carbon dioxide, which now you've gotten rid of all the carbon that started from your sugar. You're going to harvest electrons in 3 NADHs, and you're going to harvest electrons in a new type of taxi, FADH2. So FAD would be the empty taxi, FADH2 is the full taxi. These are taking electrons over to the electron transport chain, just like NADH is, empty taxi, full taxi. We're also going to make some GTP, which is really similar to ATP in your body. In fact, your body's just going to convert it into ATP anyway. So let's say we make ATP, and we're going to use up a little bit of water. At the end, we will have gotten rid of all the carbon from our glucose. So it's gone, it's exhaled, you're exhaling it right now. We're going to get a bunch of electrons. We get 1 set here in pyruvate oxidation, then we get 3 in the citric acid cycle. We're going to get even more in FADH, and we're also going to create ATP. Summarizing words, what happens? Pyruvate is oxidized into acetyl-CoA, which goes through the citric acid cycle, releasing carbon dioxide and high-energy electron carriers, and a little ATP. The cycle regenerates oxaloacetate, so it can start it again. All of those electrons are going to be used in the next step, which is the really important step where we produce the most ATP. So that's our kind of goal of the citric acid cycle, is to get those high-energy electrons. The inputs are pyruvate, acetyl-CoA, water. And the outputs are your full taxis, so you get 6 NADH from the citric acid cycle, you probably only see 3 there, but remember you made 2 pyruvates, so each citric acid cycle happens twice for every carbon, or for every glucose you ate. You're going to get 4 CO2 molecules, 2 ATP molecules, and 2 FADH2 molecules. And we follow the loss of carbons. All right, now let's take all those electrons and bring them to the big show. Here is oxidative phosphorylation, and our learning goals are to summarize what happens in words, list the inputs and the outputs, follow the flow of electrons and hydrogens, and then differentiate between the electron transport chain, oxidative phosphorylation, and chemiosmosis. These 3 words are kind of used all similarly, but they're all different, and I want us to be able to tell the difference between them. And then lastly, explain the role of oxygen. We know that oxygen is really important. We need to be breathing it to stay alive, but what does it actually do? First, let's orient ourselves. Here we have the inner membrane of the mitochondria. It's made of a phospholipid bilayer, just like your cell membrane. This is the inner membrane space, meaning that the outer membrane's up here somewhere. And this is the mitochondrial matrix, which means that this is inside the inner membrane. The inner membrane has these proteins embedded in it. These proteins together are called the electron transport chain. And over here, we have this really cool enzyme, ATP synthase, which is going to make ATP. It's going to synthesize it. So what happens? This whole process is called oxidative phosphorylation. And there are 2 parts to it, the electron transport chain and chemiosmosis. In our previous steps, we had a bunch of high energy electrons that we put into our electron carrier molecules, FADH2 and NADH. Those molecules bring those electrons over to the electron transport chain. The electron transport chain carries the electrons back and forth, so these actually move and pass the electrons to each other. And as the electrons are passed to each other, hydrogen ions are pumped from the matrix into the inner membrane space. These electrons fall through the electron transport chain, and then right here at the end, they have to wait until someone can catch them. And that someone is oxygen. I think of oxygen as like a catcher's mitt, and these as little baseballs. So you can't throw the baseball until the catcher's ready, right? So oxygen comes and grabs the electrons, taking them out of the electron transport chain. And in the process, oxygen is reduced to form water. So all of the air that you're breathing, you are taking oxygen out of it so that you can use it to accept electrons at the end of the electron transport chain, and you're turning that oxygen into water. Now, water's rad, you can use it for a bunch of different things, but that's why we breathe, which kind of seems incredible to me. So we call oxygen the final electron acceptor because it accepts electrons at the end of the electron transport chain. So this is cool. We've used oxygen, but we haven't made any ATP. So let's talk about how we do that. As these protons get pumped into the inner membrane space, they get really crowded. You create a high concentration compared to the matrix. So now we have a concentration gradient of hydrogens in the inner membrane space and a lower concentration in the matrix. These hydrogens want to follow their concentration gradient, so they're going to diffuse back into the matrix, but the only way they can get back is through this incredible protein, ATP synthase. So it ends in ACE, you know it's an enzyme. ATP synthase rotates, which is what that arrow is showing, every time a hydrogen comes through. And as it rotates, it grabs ADP and a phosphate and it crams them together and it creates ATP. So this is the exciting part. We're using the passive movement of a concentration gradient to power an enzyme that gives us energy. Chemiosmosis is this process. Osmosis was the movement of water across the membrane. Chemiosmosis is the movement of protons across the inner membrane in the mitochondria. And for each hydrogen, we get an ATP, and you can see that a bunch of hydrogen are getting pumped across. So in this step, in oxidative phosphorylation, we can make like 34 ATP from one glucose molecule. So it's really productive. This is exciting. This is the step that keeps us alive. And that's why it's so important that we breathe, because we need to keep those electrons falling through, because if they don't flow through, then we don't have a concentration gradient, and that means no ATP. All right, let's summarize in words what happens. Electrons from the first steps of cellular respiration are taken to the electron transport chain, which is a series of proteins embedded in the inner membrane. It's a physical thing. The electrons fall through the electron transport chain, and as they do, they pump hydrogen ions into the inner membrane space. The electrons are accepted by oxygen at the end of the electron transport chain and reduce oxygen to water. The hydrogen ions that are now highly concentrated in the inner membrane space follow their concentration gradient and exit back to the mitochondrial matrix through ATP synthase. ATP synthase is an enzyme that creates ATP every time a hydrogen ion flows through it. That process of hydrogen ions flowing through ATP synthase and creating ATP is called chemiosmosis. The electron transport chain and chemiosmosis are two parts of the process oxidative phosphorylation. So I think we hit most of this already. The inputs are electrons and oxygen. Outputs are water and ATP. We followed the flow of the electrons through the electron transport chain and the hydrogens through ATP synthase. We know that this is a place and this is a process, and this is a process but also uses an enzyme. And we know that oxygen is the final electron acceptor. So let's look at an overview. I like this diagram because it shows us the location. Glycolysis is happening outside of the mitochondria that you see here. Glycolysis, you take glucose and turn it into pyruvate. In the process, you take 2 electrons that you will use later in the electron transport chain. You also produce 2 ATP. Pyruvate is oxidized into acetyl-CoA, which can enter the citric acid cycle. During pyruvate oxidation, you produce more electrons that you will use in the electron transport chain later. The citric acid cycle produces a bunch of electrons. This is where we get the most of them. And it also gives us just a little bit of ATP. Then oxidative phosphorylation, including the electron transport chain and chemiosmosis, is where we produce most of our ATP, 34 ATP, and where we're going to use those electrons to power our electron gradient in our electron transport chain and chemiosmosis. So fabulous diagram there, but did you notice that it's missing something? So take a moment and think, what's this diagram missing? It's missing water, it's missing oxygen, and it's missing carbon dioxide. So really important parts of the story are just not being shown. And I understand that it would make it a more complex diagram, but still, that's why we got to be careful when we Google image search things, is sometimes we don't get the full story. So what might be a cool way to study is take this diagram, copy it, and then add the carbon dioxide, because you need to know what stages the carbon dioxide are released at. Add oxygen, where do you use oxygen? And add water, where do you produce water? Cool. So that was a lot. Hopefully you have some questions. Please email me anytime. I love talking about cellular respiration. And I hope you have a great day, and I'll hear from you soon. Bye.